Okay, now it's time to go over Chapter 10, Foundations of Genetics. When we're talking about genetics, we're talking about heredity, the tendency for traits to be passed from parent to offspring. And when I say trait, I just mean a specific characteristic determined by genes, but also influenced by the environment that can be passed down through generations. So maybe that trait is the brown eyes that I have, my brown hair, the fact that it's brown and curly, whatever your trait is that can be passed on in genetic information to your offspring. Traits are heritable features, and that just means they can be inherited. Before the discovery of DNA and chromosomes, principles of heredity were first identified by quantitative science, and that's just the science of counting and measuring. We know that quantitative is numbers and counting. Qualitative is whenever there's a range of different factors affecting something. So Gregor Mendel actually solved the puzzle of heredity very early on. This is impressive to me. He did his experiments between 1857 and 1864. And he was an Austrian priest. So Austria, which is now Czechoslovakia. And he did experiments on these pea plants. He's come to be known as the father of genetics. So he didn't really get respect for his work in his lifetime, but since he had passed away, more and more people began to realize, wow, he's really onto something here, until the point where he was eventually called the father of genetics for his groundbreaking discoveries so early on. And this all started because he was looking around the monastery where he lived and seeing all of these pea plants and their beautiful flowers. And he was wondering what affected the population of the pea plants to the point where some years there would be lots of purple flowers, some years there would be more white flowers. And he wanted to know what is determining that? Why are the flowers turning out purple or white? What's behind that? So he started running some experiments. He got lucky with this random idea being about pea plants because they're really ideal for studying heredity. There's lots of different varieties that are available, and they have lots of different easily distinguishable traits that can be quantified so that he could easily count and easily see. Also, they're really small and easy to grow, so that he could produce large numbers of offspring quickly for larger numbers of data. And also their reproductive organs can easily be manipulated, like most flowers, so that pollination can be controlled. Because if you're trying to have a specific combination of what goes in to create pea plant offspring, you don't want to have them suddenly self-fertilize. So to prevent that, he would just remove part of the flower so that the flower could no longer self-fertilize and would only be fertilized by whatever pollen he brought. So he had a very specific experimental design, and his first goal was to establish true breeding varieties of these two separate kinds of pea plants. So the first of which would be the purple pea plants and then also the white flower pea plants. By allowing plants to self-fertilize for several generations, that's how he was able to create his parent generation, which he called the pea generation. And these would be true breeding varieties. So basically, he noticed that sometimes pea plants with purple flowers would produce plants with white flowers. Sometimes they would be purple. He was trying to make sure he had one line of purple plants that only ever created purple plants. So how he did that was he took out a single purple pea plant. He let it self-fertilize over and over and over again until he finally was getting no white offspring flowers at all. And then he said, okay, I've established my parent generation. And then did the same thing for the white flowering pea plants. He then crossed the two varieties with those alternative traits of purple flowers and white flowers, and he named the resulting offspring the F1 generation, which stands for filial, like offspring, but you don't have to worry about that. We're just calling it the F1 generation. He then allowed the plants from that F1 generation to self-fertilize, and named the resulting offspring the F2 generation. So let's look at that here. Here you can see we've got our two parent generations for white flowers and for purple flowers. He got those by allowing them to self-fertilize until there were no leftover traits from either side. Once he had his true breeding parent generation, he cross-fertilized them together to see what offspring he would get. So he mixed the true breeding white flowers with the true breeding purple flowers. It turns out all of the offspring in the F1 generation were purple. He then let those flowers self-fertilize and looked at the results for the F2 generation. So in the F2 generation, we see this white flower characteristic has come back. Mendel experimented with a variety of traits, and he repeatedly made the same observations. So he did this with multiple things. He did it with seed color, seed shape, pod shape, the height of the plants, the color of the flowers, all of these different traits. He did this same repetitive process and just kept getting the same number in his results for the ratio between what was being expressed and what wasn't. It turns out for each pair of contrasting varieties that he crossed, one of the traits would disappear in the F1 generation, just like we saw, but then reappear in the F2 generation. And then he called the trait expressed in the F1 generation, like the purple flowers that we saw, the dominant trait. And he named the trait that would disappear and then come back the recessive trait. He counted the number of each type of plant in the F2 generation, and he found really consistent proportions of traits expressed in his different crosses. So it turns out 3/4 of the F2 individuals expressed the dominant trait, while 1/4 of them expressed the recessive trait. So that means we have a dominant to recessive ratio of about 3:1. It was always extremely close to 3:1. And here's an example of some of the things he was looking at. So we can see he ran these experiments with purple flowers versus white flowers, yellow seeds versus green seeds, round seeds versus wrinkle seeds, green pods and yellow pods, and so on. But in all of these, he had this same result. 3/4 of them were something dominant, 1/4 of them were something recessive, and so on and so on and so on. You can see really consistent results. Mendel reasoned that the recessive trait must somehow be hidden in the F1 generation and just not expressed. So basically, even though the F1 generation all turned out purple, the white trait is still there, just not expressed. It's hiding, it's masked. So then he allowed the F2 to self-fertilize and form the F3 generation. In that generation, 1/4 of the plants from F2 that were recessive were true breeding recessive in the F3, and 1/4 of the plants from the F2 that were dominant were true breeding dominant in the F3, and then the remaining half of the plants showed both traits in the F3. So that means he determined that that 3:1 ratio that he observed in the F2 generation is actually just a disguised 1:2:1 ratio. So basically, he thought because all 3 of these are purple, or whatever the dominant trait was he was looking at, but in this case we'll say purple, because 3 of the 4 turned out purple, we've got a 3:1 ratio. But that's not really true, because one of them is true breeding dominant, meaning it can only produce purple, and 2 of those purple flower plants still have the ability to create offspring with white flowers. So they're not true breeding dominant. So that means we have a 1:2:1 ratio. This one can only produce purple flowers. This one could potentially produce purple or white flowers for both of these. And then this one is only going to be able to produce white flowers. So we can see that here. We've got our parent generations that are true breeding. We combine them into F1, they're all purple. We let that self-fertilize, so we get F2 generation, what looks like a 3:1 ratio, but it's not, because one of them can produce only purple, 2 of them could produce purple or white flowers in the next generation, and 1 of them could only produce white flowers in the next generation. So after all of his work, he proposed 5 hypotheses to explain his results, basically. In hypothesis 1, he said parents do not transmit traits directly to their offspring. They transmit information about the traits in the form of what he called factors, which is insane because that means in 1865, this priest discovered genes, because what he's calling factors are really genes. That's exactly what he was talking about. They've been renamed since then, but every time he mentions factors, he's talking about genes. In hypothesis 2, he stated that each parent contains 2 copies of the factor, or gene, governing each trait. So the 2 copies of the factor may or may not be the same. If they're the same, we call that a homozygous inheritance. And if we notice that those are different, that's a heterozygous individual, meaning it has 2 different copies of that same gene. So basically, you can see that here. In this chromosome, you could consider that we got this gene from our dad and this gene from our mom. Maybe it has to do with eye color. So if so, they match. They're the same. This is a homozygous chromosome. So we've got possibly brown and brown eye color, or blue and blue eye color. But if those are separate, if they're different from each other, we call that a heterozygous chromosome. So here you could imagine that this would be the blue eye color and the brown eye color. Maybe only brown is being expressed, but they're both there. They were both inherited. In hypothesis 3, it states that alternative forms of a factor lead to alternative traits. And that is basically talking about alleles. So alleles are defined as alternative forms of a factor in Mendel's words. But in our current definition of alleles, we think of it like this down here. An allele is one of 2 or more, and that more could be like hundreds, 2 or more versions of a DNA sequence at a given genomic location, and an individual will inherit 2 alleles, one from each parent, for any given genomic location where such variation exists. So basically, you could think of it as the gene is eye color, the allele is for brown, blue, green, gray, or you could say the gene is hair type, the allele is thick and black and coily, or curly and brown, straight and blonde, any combination of those. But we're talking about the same gene. The allele is the difference in how it will be expressed. So here you can see an example where we're kind of highlighting that this is a heterozygous allele. You can see that this allele is dominant and this one is recessive. This one is recessive and this one is dominant, which we're going to go into more detail on. And then you can see here we have an example of homozygous alleles where these match. So whatever gene that this is whatever this gene is coding for, it's going to be in the same location, but these will match. So if it's a homozygous, we have matching alleles. Like this could be the chromosome that has purple flower, or white flower, white flower. Maybe this is the one that has one trait that could be purple, one trait that could be white. So I want to kind of highlight that when we're separating genotype from phenotype, the genotype is the organism's genetic information. So when we're talking about the gene, like this, when we're using those words of homozygous or heterozygous, that's the gene we're talking about. When we say phenotype, that's whatever the expression is of that gene. So what we can see. So for example, this homozygous dominant, dominant because it has 2 genes for purple, 2 alleles for purple. The expression, the phenotype of that, is purple flowers. Here we have a homozygous recessive gene, meaning it has 2 alleles, both of them are recessive and for white flowers. The expression, the phenotype, is the white flowers. Here's another example. So we were talking about eye color. Genotype we're talking about would be like two uppercase B's for two dominant brown eye alleles, and then a lowercase b representing the recessive blue eye alleles. So that's talking about genotype. When we mention what kind of phenotypes we have here, it's literally just brown eyes or blue eyes in this example. So phenotype, the expression of that gene. In hypothesis 4, he stated the two alleles that an individual possesses do not affect each other. In hypothesis 5, he stated the presence of an allele does not ensure that a trait will be expressed in the individual that carries it. So just because you have that allele doesn't mean it's going to show up in your phenotype, doesn't mean it's going to actually be expressed. Just like I could have a hiding allele for blue eyes, but you'll never know because the brown one is the one that's expressed. And in heterozygotes, that's often the case. Often only the dominant allele is going to be expressed. Here are some examples of some common recessive and dominant traits for humans. So we'll just do one in this summary, and you can pause and look at it longer if you want to. But one example of a recessive trait is common baldness. The expression of that gene is an M-shaped hairline receding with age. And that's a recessive trait. So if one of your parents has this trait, it's more likely that a dominant trait will overpower it because it's recessive. Dominant traits, though, like mid-digital hair, so if you have any hair in the area between your two knuckles on your fingers, the presence of hair there is a dominant trait. And that is the phenotype, the expression of that gene. So genetic traits are assigned an italic letter symbol referring to their more common form. And dominant traits will be capitalized, while a lowercase letter will represent the recessive trait. So for example, we were talking about the flower color in these pea plants. Our purple flowers, which have that dominant allele, will be represented with a uppercase P because we know that's the dominant. And our recessive allele for white flowers will be represented as a lowercase p. So it makes sense. We usually have these two alleles, one from each parent. We're going to put the dominant one first, the recessive one second. Dominant is uppercase, recessive is lowercase. So we use these things called Punnett squares to help visualize what's happening with these alleles and what allele possibilities exist in the offspring. A Punnett square basically, it's just listing the possible gametes from both of our individuals that are contributing to offspring. So we can kind of see what the possible results are. And here's an example of that. So imagine that we had our two parent generations and we ended up with all purple flowers and they self-fertilized, and now we have this generation, the F2 generation, here within the Punnett square. So up here on top is the information of one parent. On the side is the information from another parent. They'll match if it's self-fertilization. They'll also match if it's two different parents that are both heterozygotes. So what we see here is this parent has a dominant allele for purple flowers, a recessive allele for white flowers. Now we're considering how it's going to mix with the other side of our genetic information, where we also see a dominant allele for purple flowers and a recessive allele for white flowers. So when we look at this square, what color would this expression be? Purple. Hopefully you said purple, because that's right. There's no recessive allele here for white at all, so there's no chance that this offspring is going to be white. Whenever we look at these two squares, which are matching, you can see we've got a dominant allele for purple and a recessive allele for white, making this heterozygous. So the chance of these producing purple offspring is higher, but there is potential that they'll produce that white flowering offspring as well. Then we look at this area. You can see we've got the recessive allele from the parent here, the recessive allele from the parent here. That results in white flowering plants. So this 25% of these offspring will have this homozygous recessive expression where they can only produce white flowering plants. So let's look at that. We've got our Punnett square here, which is the same as the example we just saw, but with visuals. We've got our heterozygous parent here, our heterozygous alleles from the parent here, and we can see how they combine. So in this box, the offspring has received a dominant allele for purple and a dominant allele for purple. In these two boxes, the offspring has received a dominant allele from purple and a recessive allele for white. And then here you can see we've got a dominant allele from purple and a recessive allele for white. And then this portion of the offspring is going to receive the recessive allele from both sides. So that makes it homozygous recessive and only able to produce white flowers. Also, this image helps to make sense of why, when we cross true breeding purple with true breeding white, all of the offspring are purple. Because we're receiving an allele from each parent, all of them are dominant purple from this parent and recessive white from this parent. So there's no chance to get anything else because that recessive trait is going to be overpowered by the dominant trait. So that's why they all turn up purple. But then, when you go on to make another generation, and you're mixing these heterozygous alleles, that gives the opportunity for two recessive alleles to be inherited. And that's what's going to result in those white flowers coming back in the next generation. So Mendel also devised a test cross in order to determine the genotype of unknown individuals in the F2 generation. So the unknown individual would be crossed with a homozygous recessive individual, so a plant that has two recessive alleles for white flowers. And if the unknown would be homozygous, then all of the offspring are going to express the dominant trait, purple flowers. If the unknown is heterozygous, then half of the offspring would express recessive traits, like the white flowers. You can see that happening here. So here's our unknown flower. He knows it's purple. He's trying to figure out, is it homozygous or heterozygous? Does it have that recessive gene that could be carried on? Probably this is how he was testing to make sure his final self-fertilization batch generation was actually completely homozygous, because he was able to test it by doing this. So he took that unknown purple flowering plant and crossed it with a known homozygous recessive white flowering plant. He knew that if it was homozygous, if this unknown purple flower was homozygous, the result would be 100% purple flowers in the next generation, because that homozygous would be masked. Over here, this recessive trait would be masked and covered up by the dominant homozygous trait coming from this flower. But if it wasn't actually a true breeding plant that was homozygous, whenever he mixed it with a homozygous recessive plant, then he would end up with half of the offspring expressing that white trait, because this allele was brought by the homozygous recessive and masked in this this organism. So once they mix together, then we're able to get white offspring because of these alleles lining up. There's no dominance here, only recessive. Mendel's hypotheses very neatly predicted the results of his crosses, and so neatly that they have come to be called Mendel's laws. And really, we found out later on, he predicted them a little too well. The data looked a little too good. So in the 1960s, when everything was really opening up for science and studies, a lot of people looked back at Mendel's work, and they noticed something. They noticed that the numbers were just a little too perfect, And it's not usually like that in science. We know that we have mutations. We know that things change all the time and defy the common way that things happen. But we weren't seeing that at all in Mendel's numbers. So the conclusion is that he probably messed with the numbers a little bit. He left out the outliers. He made these numbers look like it happened 100% of the time, whenever it probably could not have been 100% of the time. But really, his contribution to science was still huge. And he was also a priest in the 1860s, surrounded by other priests, doing science experiments. He wasn't a scientist surrounded by scientists with the common standards we have now to not exclude any of your data, to make sure that you're not altering the conclusion, basically, that you're getting from that data. So it seems like a general consensus that he probably did exclude some of the results that didn't match the most common finding. But even so, he managed to discover the foundation for genetics. And the first law that he came up with based on his hypotheses is the law of segregation. So this law states that the two alleles of a trait will separate from each other during the formation of gametes, so that half of the gametes will carry one copy and half will carry the other copy. Mendel also investigated the inheritance pattern for more than one factor. So when crossing individuals who are true breeding for two different characters, the F1 individual that results is called a dihybrid. After that dihybrid individual self-fertilizes, there are 16 possible genotypes of the offspring. And this is what he showed with the dihybrid cross. So here you can see we've got parent generations, and we're going to be looking at two different traits. Are the seeds yellow or green, and are they round or wrinkled? You can see how they're represented here. Here's our homozygous gene for round shape and homozygous for dominant yellow shape. Here's homozygous for wrinkled shape, which is recessive, and homozygous for green, which is recessive. After fertilization, when those mix, and then we have our separation where meiosis, where we go through meiosis and chromosomes will assort independently, we'll end up with our 4 separate gametes. And these are the possibilities for how they turned out after we mixed homozygous dominant and homozygous recessive. So you can see this first one is dominant for round shape, represented by that uppercase R, and dominant for yellow color, represented by that uppercase Y. This one is dominant for round shape, recessive for color, so it's green. And this one is recessive for shape, so it's wrinkled, and dominant for color, so it will be yellow. This one is recessive for shape, so it's wrinkled, and recessive for color, so it's green. Now we zoom into this Punnett square, this dihybrid cross, to see what the results could potentially be. And you can see them here. So we've got dominant for color or for shape and color, resulting in both dominant round and dominant yellow color. This one you can see has dominant for shape, so it's round, and recessive for color, so it turns out green. This one here you can see we've got a recessive allele for shape, so it's wrinkled, and a dominant allele for color, so it turned out yellow. You get the idea. So that's how he was able to determine what the potential results would be for basing this on 2 separate traits. In Mendel's second law, he concluded that for the pairs of traits that he studied, the inheritance of one trait does not influence the inheritance of the other trait. This is Mendel's second law of independent assortment. Basically, genes located on different chromosomes are inherited independently of one another. So we know that genes are encoded in DNA, and we know DNA is transcribed into RNA. that transcribed RNA is going to be translated to produce a polypeptide, which is just a chain of amino acids. And we know the specific sequence of amino acids will determine that polypeptide shape, which is going to affect how the protein is going to function, which then in turn affects the phenotype. So it makes sense to say that changes in DNA can affect the order of amino acids in a polypeptide, and then also end up changing the phenotype of that gene. So yeah, we change the DNA, we change the phenotype that shows up. Often the expression of phenotype is not really as straightforward as it has seemed so far. So it's not always just one gene determines one thing, and if it's dominant or if it's recessive. It's more complex than that. We have characters that can show a range of small differences when multiple genes act together. This type of inheritance is called polygenic inheritance. So if you have multiple genes acting together to influence one expression, like maybe you have 4 different genes that are all contributing to what your hair type is, then that's polygenic inheritance. So multiple genes. Think poly like many and genic like genes. Multiple genes affecting one characteristic. The gradation in phenotypes that we talk about related to these things is called continuous variation. So it's not always as simple as just round or wrinkled, yellow or green, purple or white, yes or no, this or that. We also inherit characteristics that have continuous variation. One of the great examples of that is just the height. So we have this gradient of different heights that we could end up being, but they're not determined by one gene. They're determined by multiple genes. And also the environment that we're in. So this person is 6 feet tall, but maybe he wouldn't have been. What if he had been kept in a small room, not allowed to exercise, and forced to drink coffee from the ages 13 to 18? Maybe he would have been over here. Maybe he would have not made it all the way to 6 feet. So we've got multiple genes working together and a continuous variation of what that phenotype could be expressed as. It's not just yes or no. It could be many things within this range. Also, we've got discontinuous variation. So that's the other side of things. We do have genes that represent discontinuous variation, and they will deal with a few clear-cut phenotypes. So this could be like purple flowers or white flowers. Also, they are controlled by few genes, not many, and those genes will not show additive effect. And also, discontinuous variation, the genes will not be modified, or the expression of them is not going to be modified by any environmental changes. But in continuous variation, we're dealing with a whole spectrum, a whole range of phenotypes, and they're controlled by many different genes. And also, genes will show an additive effect. So, for instance, the more dark genes that we have, the darker our skin color will be. But also, in continuous variation, this expression of the gene, the phenotype, can be changed by what the environmental conditions are. So even though you have multiple genes determining your skin color, if you expose your skin to more and more sunlight, you will produce a darker skin color. And that's an example of continuous variation. So we talked about polygenic inheritance, where many genes are affecting one trait, and pleiotropic is kind of the other side of that, where we have one allele that is affecting more than one phenotype. So these effects are characteristic of many inherited disorders. Lots of diseases that people get from their parents that are passed down are from pleiotropic alleles. And examples of this are like cystic fibrosis and also sickle cell disease. You can see that represented here. So we've got one gene, the CF gene, that is producing all of these different expressions, all of these different phenotypes, basically. So this gene is going to produce faulty chloride transport channels, causing mucus to build up. And that is having an impact on the cells in the liver, the pancreas, the lungs, and the sweat glands. So it's just one gene with all of these effects. In the liver, it's going to cause blocked fat digestion, which could result in vitamin deficiency, causing night blindness, skin disorders, rickets, and bleeding. In the lungs, it's going to cause a failure of cilia to be able to flush that mucus from the lungs, which could then cause respiratory infections. And in our sweat glands, it could result in producing overly salty sweat. So maybe our body should have kept more salt than they did, but because of this CF gene and the effect of it, now we are getting rid of too much salt from our system. So that's an example of pleiotropic effects. We have one gene affecting multiple things. Also, I told you guys, the more you talk about genetics, the more complex it gets. Not all alternative alleles will either be fully dominant or fully recessive in heterozygotes. In these cases, the alleles will exhibit incomplete dominance. And that is basically going to produce a heterozygous phenotype that is in between the two dominant parents. So it's kind of creating a new possibility. Here's an example of that. This is incomplete dominance. So here you can see we've got this flower, which is a true breeding plant for red flowers. It is homozygous. Both alleles that it's bringing are dominant for red. Here we've got a homozygous parent generation that has the dominant allele for white in both cases. So two dominant alleles, it's homozygous for that white allele. The incomplete dominance is how we see the change in the offspring when those two dominant forms go to the same flower or the same plant. So in this case, instead of us ending up with 3 red flowers because red's dominant and white's recessive, they're both dominant. And since they're both there, they both get expressed and we end up with pink flowers. This is incomplete dominance. Red didn't win, white didn't win. They both showed up and we ended up with pink flowers. So both are dominant, but not completely. They're not going to win completely. They're both going to add part of the expression for that phenotype. Also, when we talk about environmental effects for these genes, temperature is a great example as well. So the degree to which a lot of alleles are going to be expressed will depend on the environment. And when we talk about temperature, we've got species like Arctic foxes, Himalayan rabbits, and Siamese cats that are all sensitive to the temperature, and that'll change their pigment production. So for Arctic foxes, they will only produce fur pigment when temperatures are warm enough. Otherwise, they produce no pigment so that they can really camouflage with the snow. Then once it gets really warm out, they start making pigment again. And that allows them to be darker and be more camouflaged in their environment. The Himalayan rabbits and the Siamese cats also have a gene that encodes a heat-sensitive enzyme called tyrosinase, and that controls pigment production as well. So the tyrosinase is going to be inactive at high temperatures and active at lower temperatures, meaning that at lower temperatures they will produce that pigment. We also have epistasis, which just refers to the interaction between the products of two genes, where one of the genes is modifying the phenotypic expression produced by the other. So, for example, in 1918, geneticist R.A. Emerson crossed 2 breeding varieties of white corn, Zea mays, the scientific name, and the F1 plants had purple seeds because of 2 genes interacting. So those genes interacted together and changed the seed color because of that interaction. This is epistasis. Another example of how that happens is shown here. So we've got an allele for no dark pigment in the fur, and the way that that is going to show up in the offspring is affected by the interactions between another allele. So when we take this allele for no dark pigment in fur and mix it with this allele, we end up with yellow fur, but brown nose, eyes, and lips. In the same case, if we have that same allele for no dark pigment in the fur and mix it with this allele, then we end up with still yellow fur, but instead we have a black nose, black lips, and black eye rims. Same thing over here. We've got an allele for dark pigment in the fur. If it's mixed with this allele, we'll end up with a dog that has brown fur, brown nose, brown lips, brown eye rims. If we mix that dark pigment allele with this allele, then we end up with black fur, black nose, black lips, black eye rims. So you can see these are how one allele is affecting another allele and how it's expressed, which is epistasis. We also have codominance, and at first look, it kind of feels like incomplete dominance, but it's different. So bear with me. In codominance, a gene can have more than 2 alleles in a population, and often in heterozygotes, there won't be a dominant allele. Instead, both alleles will be expressed. So these alleles are said to be codominant. And it sounds kind of like incomplete dominance where we're expressing both of them, but we're not creating a new phenotype. We didn't make a pink horse because we have both the red allele, the red fur allele, and the white fur allele. So we didn't end up with a pink horse. Instead, it's codominance. Both of those are expressed in their own way. We've got red fur and we've got white fur. There are also examples where multiple alleles are affecting this. So the gene that determines ABO blood type in humans exhibits more than one dominant allele. So basically the gene is going to encode an enzyme that adds sugars to lipids on the membranes of our red blood cells. And those sugars are going to act as recognition markers for cells in our immune system. So that determines what our cells say is okay and what needs to be destroyed. The gene that actually encodes that enzyme is designated as this uppercase I, and it has 3 separate alleles or possibilities. So it's got I uppercase A, I uppercase B, and then lowercase I. So which will be recessive. So different combinations of the 3 alleles could produce 4 different phenotypes, which are the blood types, A, B, AB, and O. So both IA and IB are going to be dominant over lowercase I, and they'll also be codominant, meaning if they both, if an organism, if a person, if a baby receives an allele from each parent and they have an A and a B, they will be codominant. They will both be expressed. So you can see that represented here in this Punnett square. So here we've got possible alleles from the female, and here we've got possible alleles from the male. You can see if mom had this IA allele and dad contributed an IA allele, our blood type will be A. If mom contributed an IB allele while dad contributed an IA allele, because of this being codominance, both will be expressed and the individual will have AB blood type. Here you can see this blood type is still just A because this is recessive and will not impact the blood type. But the only way to end up with O blood type is to have that allele from both parents. You have to receive the recessive allele from both parents. Otherwise, any A or B that shows up is just going to take dominance over that recessive allele. And then, so like this one here, yes, that, like this one here, yes, that I is present, but it's still B blood type because that B is dominant. So we also talk about the chromosomal theory of inheritance, and this was first proposed in 1902 by Walter Sutton, and it's supported by several pieces of evidence. So first, similar chromosomes pair up during meiosis. That's one piece of evidence supporting it. Next, reproduction involves the unions of egg and sperm, and we know each gamete contains a copy of the genetic information, and since sperm have very little cytoplasm, the material contributed probably has to reside in the nucleus. So this theory is basically stating that chromosomes both segregate and assort independently during meiosis, similar to the genes in Mendel's model. A potential problem with the chromosomal theory of inheritance is that there are many more traits that assort independently than there are even chromosomes. But experimental study of the fruit fly, Drosophila melanogaster, by Thomas Morgan, provided confirmation of this chromosomal theory of inheritance. It also provided some interesting information about sex chromosomes. So, in 1910, Morgan discovered a mutant male fruit fly who had white eyes instead of the typical red eyes. And he was trying to determine whether this trait would be inherited using that Mendelian pattern. So when you hear Mendelian pattern, just think like Gregor Mendel. That's what it means, using that same experiment design. So he crossed a mutant male with a normal female, and as predicted, the eye color segregated, and all of the F1 individuals ended up having red eyes. But in the F2 generation, he noticed the white eyes were only showing up in the males. So Morgan determined that sex was the key to explaining the results of his cross, these kind of unexpected results. Basically, in most organisms, just the same as the fruit fly, we will have a sex chromosome that determines whether we are female or whether we are male. The females will have an XX chromosome, and the males will have an XY chromosome, meaning they have one chromosome with an allele, with an X allele and a Y allele. So Morgan reasoned that the white-eyed trait has to reside only on the X chromosome. So both females and males have the X chromosome. He thinks that this trait is carried on the X chromosome, but because of the combination with that Y chromosome, that's why the red color is able to be, that's why the white color is able to be expressed in the eyes of the male fruit flies. So this is a trait determined by a gene on the sex chromosome, and that means it's a sex-linked trait, meaning of all the chromosomes we have, this one is determined by the sex chromosome. And Morgan's experiment demonstrated the chromosomal basis of that sex linkage. So basically you can see here, he's got a male fruit fly and a female fruit fly. And the male fruit fly has this Y chromosome and an X chromosome. That X chromosome has the white eye allele. And this female, we've got two X chromosomes that both have the red eye allele. When we go on to the next generation, the F1 generation, then we have a male that is showing red eyes and a female that is showing red eyes, carrying that white allele all the same, even though she's female, but it's not being expressed because that red eye on the X chromosome is dominant. So they could end up, the males could end up with red eyes if their Y chromosome mixes with an X chromosome that has the dominant red eye allele. But if their Y chromosome mixes with one that has a white eye allele, then that's what's going to be expressed, like right here. And the reason that this is happening in the males and not really the females is because even when the females receive that white allele for the white eyes, they have the other X chromosome with the dominant red eye allele, so that just overpowers the expression of that phenotype. So that's why we do have males and females with red eyes, but we're only witnessing males with the white eyes. So Morgan's demonstration of sex linkage in the Drosophila confirmed the chromosomal theory of inheritance, which is just that Mendelian traits reside on chromosomes. And it also explains why Mendel's first law of segregation works, because traits will assort independently because chromosomes assort independently. So it makes sense if the traits are on the chromosomes and the chromosomes assort independently of each other, then so too do the traits. So linkage is defined as the tendency of close together genes to segregate together. The further that two genes are from each other on the same chromosome, the more likely crossing over is to occur between them. And the closer that two genes are on the same chromosome, the less likely that crossing over will occur between them. So the first one, this would lead to independent segregation. The second one, these genes almost always segregate together, and so that makes sense that they would be inherited together. You can see that happening here. As crossing over happens, you can see we've got this allele here and this allele here. Both are for flower position. These alleles are for pod shape. These are for plant height. So if we look back, the further two genes are from each other on the same chromosome, the more likely crossing over is to occur between them. The further they are apart, the more likely crossing over will happen so that they become combined. If they're really close together, when crossing over happens, they're still going to be together because this is not going to cut right here, most likely. It's probably going to be further away from that. So that's all that we mean whenever we're talking about the differences in likelihood of whether or not chromosomes and genes will be inherited together. Also, when we're talking about our chromosomes, we know each human somatic cell, so our normal body cells, has 46 chromosomes, which in meiosis is forming 23 pairs. So 22 out of those 23 pairs are going to be perfectly matched in both males and females, and they're called autosomes. But one pair of those 23 are called sex chromosomes. That's what we just talked about with Drosophila. So the females will have XX chromosomes, males will have XY chromosomes. And the genes on that Y chromosome are actually what determine maleness. So sometimes we have errors that occur during meiosis. And one example of that is non-disjunction. Non-disjunction is the failure of chromosomes to separate correctly. And that can happen either in meiosis 1 or meiosis 2, but it's happening in meiosis. If the chromosomes don't separate correctly during meiosis when we're trying to form our gametes, this could lead to aneuploidy, which is an abnormal number of chromosomes. Also, most of these abnormalities really have a severe impact on the person. So they can cause a failure to develop or an early death before adulthood. It doesn't go well for us. We're really made to function with our specific number of chromosomes. If we have too many, if we have too few, that has a really extreme impact on our lives. So in contrast, individuals with an extra copy of chromosome 21, or more rarely, chromosome 22, can survive to adulthood. However, these individuals have delayed development and intellectual impairment. So Down syndrome is something that is caused by having an extra copy of chromosome 21. That's one example of the changes that can happen whenever we have one extra chromosome. So you can see that happening here. We've got non-disjunction happening in anaphase 1. You can see this chromosome was supposed to be pulled to this side. Instead, it went to this side. So now this cell is going to have too many chromosomes. This cell is going to have too few chromosomes. So now we've got 4 separate gametes that are not functioning the way that we are designed. So basically, these are both 2n plus 1. You can see 2n and plus 1. And then this one is n minus 1, is how we would represent that. So when we talk about Down syndrome and how that is an example of when we've received one too many chromosomes. You can see that here. This is the karyotype for this person with Down syndrome. And you can see we've got 3 chromosomes in the 21st position where we should have only 2. The incidence of Down syndrome per 1,000 live births is shown here. And this is a correlation between the mom's age and how often the offspring will have Down syndrome. You can see that as mothers age and their eggs age as well, we have an increased incidence of this happening, where we're having more and more errors in that meiotic process when we're, fertilizing and creating offspring. So that is something that does affect offspring and how often they inherit an extra chromosome. It's not 100% though. You could still have a child at 18 years old that has Down syndrome, and then have 4 more children through the rest of your reproductive period, and none of them have Down syndrome. So it's not 100%, it just is an increased likelihood. Non-disjunction can also affect the sex chromosomes. So non-disjunction of the X chromosome usually creates 3 possible viable conditions. And when we say viable, we mean it still works. It's still able to function and produce offspring when that reproductive cycle is possible. So these 3 possible viable conditions are like a triple X, which is where you have 3 X chromosomes. And usually these offspring will be girls that are taller than average, and other symptoms can kind of vary. There is also the possibility for an XXY male, so a male that has an extra X chromosome. And in most cases, these men will be sterile with many female characteristics, slightly effeminate, and diminished intellectual capacity. So this is called Klinefelter syndrome. And then another syndrome where we have an XO female, which will be a sterile female with webbed neck and diminished stature. That's called Turner syndrome. So these are examples of non-disjunction, where things didn't quite go to plan with our separation of chromosomes. So you can see that happening here. We've got XX from this female parent. Because of non-disjunction, we end up with XX and O because of, and then we've got our X and Y from our sperm. So as a result, you could see here we have too many chromosomes. Here we have not enough chromosomes. When we mix that with the chromosomes from the male, we could end up with 3 X's, which is triple X, which is triple X in the female. We could end up with Turner syndrome, where we have not enough chromosomes and 1 X from the male. We could end up with Klinefelter, where we have that extra X chromosome, and this is a non-viable embryo because we have no X chromosome combined with a Y chromosome from the male. Non-disjunction of the Y chromosome will also occur in some cases. It is more rare and usually has less of an effect, but in such cases, YY gametes will be formed, leading to the XYY in males. And these males are really typically fertile and have a typical appearance. And the reason that probably is because really the Y chromosome is a very specified chromosome. Its major function is to determine maleness. So there are not a lot of other genes and there are not a lot of alleles that could be really impacted by having an extra Y chromosome. It's the least harmful extra chromosome you could possibly have. So that's why these males are typically fine, and this is kind of the best case scenario for non-disjunction, is to have an extra Y chromosome. To study human heredity, scientists examine crosses that have already been made, and they identify which relatives exhibit a trait by looking at family trees, which are pedigrees. When we're talking about it scientifically, that's a pedigree. And really what we're looking at is the genes that you've received. So often, one can determine whether a trait is sex-linked or autosomal, and whether the trait's phenotype is dominant or recessive by looking at these pedigrees. So here's a pedigree of albinism. And in this situation, males are represented as a square and females are represented as a circle. We have the affected parents have their filled in shapes. So these would be parents that do have albinism. And then these are parents that have that allele for albinism, even though it's not being expressed. So we call those carriers. They're heterozygous. They have one allele for normal pigment production and one allele for albinism. So you can see that happening here. And then these are unaffected males and females. But if we look, we can look at somebody that's affected. So all 4 of these children have albinism. We look up, we see both of their parents have albinism. So that kind of makes sense. But then when you consider this child and this child, both had parents that had that carrier allele for albinism, and that's what contributed to them ending up with albinism while their siblings didn't. So this is an example of how we can use pedigrees to trace back the inherited traits through our family tree. Here's another pedigree on color blindness. So again, you can see this person is colorblind. This person is carrying the trait for colorblindedness. And if we go up, we can see where that started here. So this is a male that was colorblind and a female that had carrier trait for colorblindness. Their offspring resulted in 1 colorblind child, 3 carriers, and 2 that were unaffected. We go down, we could look at this branch to see this person is affected, this person is not colorblind, this person is a carrier for colorblindness, and in the offspring that was expressed. So this is an example of pedigrees, and they're very useful in the field of genetics. We also have accidental changes in genes that happen, and we know those are called mutations. So a mutation occurs only rarely and will almost always result in recessive alleles. So typically they're not eliminated from the population because they're not usually expressed in most individuals, the heterozygotes. In some cases, a particular mutant allele has become more common in human populations and produced harmful effects called genetic disorders. So when we're talking about genetic disorders that are passed down through your family line, that is a mutation that was likely recessive and causing no impact, like for a carrier for a really long time, until more and more of that allele was passed on and then combined with another recessive allele to the point where it could be expressed in the offspring, and then passed on enough that it became noticeable to us as a species, that we're like, oh, this is something that keeps happening. This is a genetic disorder. Hemophilia is an example of a recessive blood clotting disorder, and some types of hemophilia are sex-linked. So the royal hemophilia with Queen Victoria actually arose due to that mutation carried by Queen Victoria. So they talk about how a lot of the royal bloodline is hemophilic. Well, that all started with, is traced back to this Queen Victorian bloodline, all the way back here. So she was a carrier for hemophilia. As we look down and see the different people that came from her bloodline, we can see Alice was also a carrier and Leopold was hemophilic. When we look at their offspring, we can see Princess Alice was a carrier. Viscount Tremation, what a name, was hemophilic. And this is an example of just another genetic disorder that can be passed on through your genes. Sickle cell is also a recessive hereditary disorder. And this is a really big deal. It affects individuals that are homozygous recessive for that mutated gene. And then if you are homozygous recessive, you will produce a defective version of hemoglobin, which is very important. We need hemoglobin very much. The hemoglobin is what sticks together and forms rod-like structures that are then going to produce a really stiff red blood cell with the wrong shape. So because of that, the cells won't be able to move through the blood vessels easily, and they tend to clot, which causes intermittent illness and shortened lifespans. Because if your blood is clotted and suddenly your blood can't get through, or even if it's not completely closed off, but you've increased the pressure in your system, that's a problem. It's not good for your system. So here you can see those blood types. This is this is a sickle cell disease. So you can see our normal red blood cell looks like this, but in sickled red blood cells, we have this shape, which you can see is the perfect kind of shape to lock together and form clots. And that's exactly what happens. So here you can see we've got a homozygous person that is homozygous for sickle cell. They have the disease. And they're having offspring with somebody who is homozygous for the normal allele, so their red blood cells are unaffected. All of the offspring are going to receive that sickle red blood cell allele from this parent and that normal allele from this parent, meaning they've all become carriers. And if they combine their allele on their side, well, let's just look at one for a better example. So we go here. If this one offspring combines their carrier sickle cell allele with another person who has a carrier sickle cell allele, or who just has both alleles and is like fully shaded in, either way, that can produce offspring that have the disease. And individuals that are heterozygous for the sickle cell mutation are generally indistinguishable. So they're not expressing anything different from the people that are homozygous for the unaffected allele. However, those heterozygous individuals, some of the red blood cells do become sickled when oxygen levels become low. So they are still slightly affected. And this may explain why the sickle cell allele is so frequent among people of African descent. The presence of the allele increases resistance to malaria infection, which is a common disease in Central Africa. So when we look at the incidence of sickle cell alleles being passed on in Africa compared to the incidence of malaria, we notice a correlation. This isn't maybe the best map to show that, but it is from the book. But there is a correlation between how often sickle cell alleles are passed on and how common malaria is in your area. Also, there's Tay-Sachs disease, which is another disease caused by a recessive allele that can be passed on through genetics. And it's an incurable disorder where the brain is actually deteriorating. So people that have this, they suffer and they rarely live beyond 5 years of age. It really causes problems. The brain is basically just kind of eating it. The cells are eating themselves, and the brain is deteriorating very quickly for a very short lifespan. There's also Huntington's disease, which is a genetic disorder caused by a dominant allele, not a recessive one. And it will cause progressive deterioration of brain cells as well. So basically, every individual who carries the allele is going to express the disorder, but most persons will not know that they are affected until they're about 30 years old in Huntington's disease. You can see here that it is a dominant genetic disorder, and that the older you get, the more likely you are to notice that expression of that gene. Genetic counseling is actually the process of identifying parents at risk of producing children with genetic defects. And so they're basically going to run a karyotype and look at these different possible alleles that you're receiving and determine the genetic state of those early embryos before the baby's even been born. This genetic screening can allow prenatal diagnosis of really high-risk pregnancies. And two examples of that are amniocentesis, which is whenever the amniotic fluid is sampled, and then isolated fetal cells are going to be grown in culture and analyzed. And this happens while the fetus is being observed with ultrasound. There's also the chorionic villus sampling, which is when fetal cells from the chorion and the placenta are removed for analysis. So in both of these cases, the goal is to determine whether or not the child is going to have a severe disease that has been passed on as a genetic disorder. Here's an ultrasound view of a fetus that would be kind of the view that would be happening during that amniocentesis. Genetic counselors do look at cell cultures from either amniocentesis or chorionic villus sampling. The chromosomal karyotype is used, and analysis can reveal aneuploidy, so we could tell, are there extra chromosomes, or are there really big changes to the chromosomes. Also, enzyme activity we can test. So in some cases, it's possible to test directly for the proper functioning enzymes associated with different genetic disorders, and then those could be kind of a clue. And then also genetic markers. So we're able to test for the presence of mutations at the same place on chromosomes where disorder-causing mutations are usually found, which is amazing. We're literally able to go in and look and say, right here, specifically in this spot on this gene, what do we see, and are we finding any mutations? And that's chapter 10. So that's all for chapter 10. Thanks for listening. I'll see you guys soon for chapter 11.